Hi, I'm Karen Israel. I'm a field application scientist with Illumina, and I'm going to be giving you a brief overview of the Illumina sample prep process. This overview will be followed by a presentation by Mara Costello of the Broad Institute, who will provide details on tips, tricks, and optimizations the Broad uses in their high throughput laboratory. Let's begin. Sample prep is the first step in the Illumina sequencing process. Library preparation begins by taking our template of interest, fragmenting it into small pieces, and then ligating on Illumina-specific adapters to create a library. These adapters are complementary to surface-bound oligos on our flow cell and enable us to both hybridize the library to the flow cell and amplify the fragments using our bridge amplification process. Once we have clusters on the flow cell, we can move the flow cell to our sequencer and sequence it using our reversible dye-terminator chemistry. The Illumina sample prep process is suitable for many types of input material. We prepare nucleic acid for sequencing by fragmenting, whether mechanically or enzymatically, creating its double-stranded DNA and flanking it with the Illumina adapters. Regardless of type of library we're creating, the preparation ends with the same general template structure, which is to have that double-stranded template with our adapters on either side. The variables in this process, other than the type of material you're starting with, are the size of the insert you put in, whether you're closely monitoring that insert size for doing assemblies, or whether it could be a bit looser for various mRNA counting applications. The type of adapters we put on, essentially if we would like to do single read, just single-stranded sequencing, or if we would like to get both strands of the library fragment, and whether or not we add multiplex indexing tag on there so that we could have multiple samples sequenced within the same lane, within the same flow cell at the same time. This slide shows a variety of the materials that can be used as starting input for the Illumina sample prep process. The most common are probably the genomic DNA and mRNA, but we also provide protocols for small RNA, chip sequencing, and other applications. I'm going to start by going over the genomic DNA protocol in more detail. This is our most popular protocol. It's used most frequently by our customers, and it's the basis for all of our other protocols. They may have different upfront steps for collecting material or fragmenting material to put into the library construction process, but the heart of the protocol remains the same for most of these applications. The steps of the sample prep process are shown below, and each step will be gone through in more detail on the following slides. But just a brief overview is that we start with purified genomic DNA and quantitate that and make sure we've got a good quality material going into our sample prep process. That genomic DNA is then fragmented. We repair the ends and phosphorylate the ends. We add an A overhang to the 3' ends to accommodate our Illumina adapters, which have a 3' T overhang. Then we're able to purify on gel to remove any unused adapter or fragments that are the wrong size, and put that into a PCR enrichment to amplify fragments that have adapter on both ends. And after that, we're left with slightly amplified library ready to go onto the Illumina flow cell. I'm going to start by talking about sample material input. I think it goes without saying garbage in, garbage out. So if we don't start off with a high quality template, or if we don't exactly know how much we're starting with, then at the end of the process we may not have as good a library as we could have. So Illumina suggests several methods for quantification and quality control. Among these are the nanodrop, the picogreen assay, and the qubit. Nanodrop is a pretty simple spectrophotometric reading of the sample, and it has its strengths and weaknesses. It is very easy to use, but it's easily influenced by a DNA reading could be easily skewed by RNA contamination, or the presence of certain salts can falsely inflate the yield that you have. Picogreen is a little more sensitive. It's specific for double-stranded material, double-stranded DNA, but it can also be influenced by contaminants such as alcohol left over from DNA extraction process, or certain salts that may also carry over through a extraction of DNA from original material. So I like to compare and contrast the two of them. It's also a pretty good will give you a better idea if you run your sample out on a gel quality-wise to let you know you have good intact DNA. And if you can compare those results with nanodrop reading and picogreen readings, you can get a fairly accurate quantitation of where you're starting with your template. Other methods for quality control would be the Agilent bioanalyzer. We find this especially helpful when working with RNA to check the integrity of the starting material, or from the time a RNA sample passes from the investigator to your lab, it may have gone through some degradation. So it's good that before you start making your libraries to give some of these good quality control checks. So getting into the actual Illumina sample prep process, we'll start by fragmenting the DNA into small pieces. The sample prep kit that Illumina provides gives you several nebulizer units for fragmentation of the DNA. The nebulizer is very inexpensive. It's already included in the kit, and it works pretty well if you have a lot of starting material to work with. But it does pick up the sample and aerosolize it, and some sample can be lost in the aerosolization. So people starting out with small amounts of template or very precious samples, this could be a little bit tricky. So the Broad will talk about in their part of the talk some of the methods that they've developed for more consistent shearing or sharing of samples that are more precious, where they don't want to lose as much sample and give a more consistent result overall. I think the basic method of Covaris, which is the sonic shearing, Mara will go over in more detail. Other methods we can employ would be sonication, hydroshear, which is just syringe-based mechanical shearing of the DNA, and enzymatic fragmentation would be more of a process we'd use in some of our other library prep techniques. The bioanalyzer trace below shows the results of common nebulizer shear using our kit. This chart shows various methods that can be used for fragmentation in the Illumina sample prep process. The most common methods for DNA would be the nebulizer and the Covaris, whereas for mRNA, a chemical fragmentation would be used. For the mate pair process, we would like to produce some larger size fragments, and we'll discuss that protocol in more detail later, but a fragmentation method like the hydroshear mechanical shearing helps us to produce those larger fragments. Each fragmentation method has its benefits and limitations. They are specific for the application, and the ones that would be right for you would depend on your desired end product. Once we have our fragmented DNA, we move on into the end repair process. In this section, we're going to blunt the ends and phosphorylate them. We perform the end repair using T4 DNA polymerase and Clinal, and phosphorylate the ends using T4 PNK. It's a simple enzymatic reaction taking about 30 minutes to complete. After end repair, we'll add an A overhang. In this process, we add an A base to the 3' end of the blunt phosphorylated DNA fragments. This A addition prevents the formation of concatemers of the template itself, and also helps us when we go to ligate on the adapters to direct the orientation of the adapters and prevent them from the adapters themselves from dimerizing. We do this with a mixture of DATP and Clinal exo with the 3' to 5' exo minus activity removed. Now that we have our A overhang, we can add on the Illumina adapters. We use the same library structure regardless of application. The single-read libraries are made to be sequenced in only one direction, and as can be seen in the image at the top, we only have a sequencing primer site attached to our P5 adapter. Paired read expands on that, and we also include a second sequencing primer site on the P7 adapter so that the insert can be sequenced from either end of the fragment. The last type of library is the index pair read, which has an optional fixed base pair tag between the read 2 primer and the P7 adapter that can be customized. Regardless of the type of library, it is important at this point to minimize the amount of adapter dimer that could possibly be created, and thus it's important to observe the 10:1 molar ratio of adapter to genomic DNA when making the library. I'd like to stop for a minute and talk about different library types and when to use them. The single-read libraries are made for unidirectional sequencing and can be put on single-read flow cells only. These are really good for counting applications, mRNA, ChIP-seq, and low-coverage resequencing projects. The paired end libraries have a little more utility in that you could sequence them in just one direction or bidirectionally to form paired reads. These libraries can be put on paired end flow cells for the most utility, or on a single read flow cell if you only needed unidirectional sequencing. Most of our applications are going on paired end flow cells to make paired reads. They're very easy to tailor your insert size and distribution per project. We're able to achieve tight size distributions for assembly projects, structural rearrangement, detection, and a variety of other tasks. And if you're a little bit less finicky, just doing some resequencing or needed some high coverage, you could also make some wide distribution libraries for paired end sequencing. The final library type we'll go into a little more detail in our additional applications to follow these slides. It's the Multiplex paired end library. These again can be sequenced uni or bidirectional, and the big benefit here is it will allow you to put multiple libraries, especially ones you need small amounts of coverage on, small genomes, have multiple libraries per lane. Illumina has 12 index tags that are available. They're introduced using PCR, and if you use 12 unique index tags in every lane of the flow cell, the 8-lane flow cell, you can have up to 96 libraries per flow cell. I frequently get questions regarding a library's compatibility with our flow cells, so I thought I'd take a minute to go over the topic in more detail. Pictured below are schematics of the surface-bound oligos on both our single-read and paired end flow cells. The main difference between the two are the cut sites and the methods we use to linearize the strands for sequencing. The single-read protocol only calls for sequencing off the P7 strand. During linearization, we cleave off the P5 strand using periodate to specifically target a modified base on the flow cell-bound oligo. This action destroys the P5 oligo and thus would prevent any possibility of resynthesizing that P5 strand for further sequencing. For the paired end protocol, we want to be able to differentially sequence both the P5 and P7 strands. We use an enzymatic linearization to selectively target and linearize one strand or the other. First, we use LMX1 to cut at the P5 oligo, and later with LMX2 we linearize the P7. It's important to note that linearizing the P5 strand leaves a majority of that P5 oligo intact, and that enables us to be able to resynthesize that strand later for the reverse read of the paired end sequencing. The additional bases on the oligos necessary for the linearizations on the paired end flow cell means they're only on the paired end libraries and flow cells, and so paired end libraries can be successfully hybridized and amplified on either paired end or single read flow cells. However, these extra bases on the paired end flow cells would mismatch with a single read library because they kind of line up with the insert, and that would prevent amplification. So long story short, with paired end libraries like our universal PHYX, they can be used on either flow cell type, but single read libraries must be used specifically on a single read flow cell. Now that we have our adapters ligated to our inserts, we're going to do a size selection on gel. This is going to enable us to choose our library size and to also help us clean out any excess adapter that may be left over from the ligation process. The gels on the right show some examples of cuts made at Illumina to make R&D libraries. Once we've purified our library from our gel slice, we're going to do PCR to selectively enrich for DNA fragments with adapter at both ends. This is also going to add some additional sequence that will help our fragments hybridize to the flow cell, and it will amplify the amount of DNA in the library. We do this using the high-fidelity enzyme called fusion. At the end of the process, we want to do some QC on our library to verify the size and the quantity of the library. We recommend using the nanodrop or qubit picogreen to estimate the quantity, and QC by Agilent bioanalyzer to check the quality. Here are some example output traces from the Agilent bioanalyzer showing both a 300 base pair library and a 600 base pair library. I'd like to take a minute to go over a few of Illumina's other DNA applications, the first being multiplexing and the second being our large insert mate pair libraries. Multiplex paired end libraries, also known as our index libraries or barcode libraries, are made using a special 3-primer system. The process is very similar to creation of our paired end libraries, but we add this additional primer 3, which contains an index tag. Illumina has 12 unique index tags that can be used to create these libraries. Multiplex paired end libraries have a special sequencing process that parallels the traditional paired end sequencing protocol. We start by adding the read 1 primer and sequencing down 36 to 100 base pairs into the DNA insert, at which point we use the paired end module to strip off the read 1 primer using sodium hydroxide and add in an index sequencing primer that lands adjacent to the index tag. We can then sequence 6 base pairs into the index tag and then resynthesize the strand for read 2 as we would for a traditional paired end read. From that point, we're able to put on a specialized read 2 sequencing primer, which is the opposite of the index sequencing primer, as you can see in this image, and sequence down into the P5 strand to get our read 2. Long insert mate pair libraries are a specialized type of paired end library. This allows us to explore gap sizes of 2 to 5 kbs, which is very useful for whole genome shotgun assembly and some of these structural chromosomal arrangements that investigators would like to explore. It's very difficult to form clusters using a library with insert size of 2,000 to 5,000 base pairs, because this, of course, will create super large clusters and won't amplify very efficiently. So the strategy that Illumina uses is to fragment the DNA into large pieces using hydroshear, mechanical shearing, biotinylate the far ends of those pieces, and then circularize them. So we get this biotin junction where these very distant ends come together. Then if we fragment the circularized DNA, we can specifically pull out that biotin junction using streptavidin beads and flush away all the rest of the fragment from in between the ends of the large insert. This junction piece can be treated then like a normal genomic DNA insert, where we can do the end repair, add the A tail, put on our adapters, go through gel purification and PCR enrichment. And then when we sequence this fragment, the read 1 will come from one far end of this 2 to 5 kb original fragment, and the read 2 will come from 2,000 to 5,000 base pairs away from it, which is a very interesting strategy for sequencing some of the projects where you need more assistance on the assembly for the structural rearrangements or help connect contigs. Next, we're going to talk about RNA applications. Illumina sells 2 kits for RNA applications. One for just mRNA and the other for small or microRNA sequencing. We'll start by talking about the sample prep process for mRNA. The process is very similar to our genomic process, but we need to start with a few RNA purification steps in advance. We'll take total RNA and we'll pull out the mRNA by using magnetic oligo DT beads to grab the poly A on this mRNA specifically. Once we've isolated the mRNA from the total RNA, we can randomly fragment it and randomly prime to create cDNA. We go through a standard second-strand cDNA synthesis, and from that point we've got a double-stranded DNA template that we can treat just like we do our genomic sample prep library or sample libraries. We'll ligate on the sequencing adapters, size select on the gel, and enrich with 15 cycles of PCR. And from there we can go on into sequencing and do a variety of counting applications and other mRNA-specific tasks. The final process we will talk about is the small RNA or microRNA sequencing sample prep protocol. This is one of our newest protocols and has a very unique approach for the sample prep process. There was a previous incantation of this protocol which involved many, many days and many, many gels and lots of processing. It would start with total RNA and you'd need to isolate specifically the microRNA from that and then go into all the various steps of the sample prep process from there. It was very labor intensive. This new process takes advantage of the 3' hydroxyl groups that are formed when pre-microRNAs are processed by dicer-like enzymes before they become mature microRNA. MicroRNA has this unique feature of having that 3' hydroxyl group in the pool of total RNA. We can start with total RNA and by adding in a special 3' adapter, which is pre-adenylated. We do the addition of this adapter with a mutant ligase, and we don't put any ATP into the reaction. So this adapter catalyzes its ligation specifically to the ends of the microRNA only. And from there, we can put the 5' adapter onto the other end through the standard process and perform RT-PCR to create our double-stranded cDNA library in this case. So it's a very nice approach. It really shortens the protocol down from this 4-day process to a 6-hour library prep time with only about 2.5 hours of hands-on time, and no need for purification of the microRNA from the total RNA. And because we've cut out a lot of those gel steps, this has really reduced the amount of input that we need to have at the beginning of the process. So we've already discussed some of the best practices for sample preparation in talking about preparation of genomic and DNA and RNA libraries. But to summarize, it's a good idea to be familiar with the protocol before starting it. A lot of these sample prep processes are pretty lengthy, and it's important to have all the user-supplied reagents and equipment available ahead of time. It's also vital to start with the highest quality starting material you can, to QC it before you start, and QC after, especially if you're using RNA to start. That little tip of using the bioanalyzer to detect the quality of the product before it's going in can save you a lot of process time and headaches. To use the proper material handling techniques, particularly to prevent cross-contamination or just contamination and degradation of your library, and that's particularly important for RNA application. Using a positive control in your library construction process can be very beneficial to troubleshooting downstream when and if something goes wrong in the library process, or when you see sequencing results downstream that don't quite make sense. It can be very helpful, and I think Mara will discuss more of that in her part of the presentation. And also just that final quantitation and QC of the final library is essential for cluster long quality and sequencing success. And with that, I'll hand it over to Mara to tell you more about how they've streamlined things and optimized things at the Broad Institute. Thank you.